Hello and thank you for joining us today for another TissueGnostics webinar. My name is Carly Briffa, and today we're joined by Dr. Livia Sima from the Molecular Cell Biology Department at the Institute of Biochemistry of the Romanian Academy to talk about tissue quantification of phospho-γH2AX foci in co-cultured cells exposed to radiation. Thanks for joining us today, Livia.
Hi, Carly. It's very nice for me to have the opportunity to present some of our TissueFAXS applications to the main users. Today, I will present the quantification of phospho-γH2AX foci in co-cultures of cells exposed to flash radiation. And for this, I will make a step-by-step presentation from the initial observation of a phenotype of DNA damage in melanoma cancer cells to step-by-step instructions on how to do the gating strategy and perform the image segmentation to obtain, finally, data that can be analyzed statistically. So as most of you know, cancer treatments are varying, and depending on the progression stage of the disease and the type of cancer, the patient is treated either by surgery to remove the tumor or by different treatments that aim to kill the cancer cells in the tumor and to prevent the tumor from progression. One of these therapies is the radiation therapy, and from this type of therapy benefits more than half of the patients with solid tumors. Usually for this type of therapy, the radiobiologists use X-rays, and this is an ionizing radiation that have an impact on the molecules inside the cell. Mainly the DNA is damaged. There are double-strand DNA breaks that are produced, and also there is oxidative stress inside the cells. Depending on the dose used, this DNA damage can be either repaired and we have a reduced clonogenicity of the cells after this treatment, or if a higher dose is applied, then the DNA damage is produced irreparably and the cells cannot exit mitosis. So conventionally, there are therapies that use 2 grays over a period of 5 to 7 weeks to treat patients. And this therapy also has some unwanted side effects. It produces fibrosis, it produces vascular collapse, and the patient's quality of life suffers a significant dropping. Therefore, in the last 10 years, a new type of ionizing radiation therapy was proposed, which is called FLASH. And as the words name it, flash therapy signifies a very short pulse of ultra-high energy and dose of radiation applied to the cells or the tumors. We have a new direction in our laboratory that is in collaboration with the National Institute for Lasers and Plasma Physics, where there is a facility of a high-power laser that produces energy up to 1 petawatt. Here, the laser is directed towards a jet of gas, producing electrons with very high energy. This facility is similar to the one in Berkeley National Laboratory in the USA, and these facilities are very few worldwide. So we are trying now to understand at the cellular and molecular level how this flash radiation affects cancer cells. The first paper that was published in mouse animal models was in 2014 that showed an advantage of using ultra-high dose rate flash in treating lung tumors, where the authors obtained the same benefit in shrinking the tumors, but without the lung scarring that is observed with conventional X-ray radiation. As we can observe here in the tissue sections, as well as in the survival graph, the animals that are subjected to this type of treatment show no complications up to 20 gray, as compared to the ones that suffer severe lung fibrosis after being irradiated with 15 gray in conventional setup. Also, in ovarian cancer, we have an example where authors have used the flash and conventional similar dose of 16 gray to treat non-tumor bearing mice to show that this type of new radiation has the ability to spare the normal tissues from the killing effect of radiation. So we can observe here at the 96-hour post-radiation that the animals that were subjected to flash radiation have the capacity to regenerate their intestinal tissue, while those that have been subjected to conventional radiation have a difficulty to regenerate the cells. After 12 weeks post radiation, the animals that were subjected to flash have a similar tissue structure in their gastrointestinal tract as the ones that were not irradiated. Also, the animals that were exposed to flash show a higher survival than the ones that have been subjected to conventional X-ray radiation, and also recover from the decrease in body weight that is observed with the exposure to X-ray conventional radiation. So therefore, this flash radiotherapy represents a new cancer treatment modality with high potential to treat deep-seated tumors, to reduce tissue toxicity while retaining the killing effect that is wanted on tumor cells. So the main characteristic of this flash therapy is a very high energy in a very short time applied to the tissues. So our workflow consisted of using co-cultures of melanoma and melanocyte cells to address the problem of the sparing effect that we want to obtain using flash therapy as opposed to conventional X-ray radiation. For this, we have co-cultured cells in a slide flask that is a very tight sealed container able to be used for 15 minutes irradiation in an incubation chamber in a flash setup as compared to a conventional X-ray radiation facility. So after exposing cells to radiation, they are then recovered for 30 minutes at 37 degrees and then fixed for analysis. And here is where our TissueFAXS analysis has the most importance, because the main assay that is used in this field to validate the effect of radiation is the quantification of foci inside the nucleus. And for this, we have used an antibody against the phospho-γH2AX that is expressed inside the nucleus. And we can see here the red signal in irradiated cell as compared to cells that were not exposed to any type of radiation. After the staining, we use the TissueFAXS system to automatically scan the slides, and then we use TissueQuest to quantify the number of foci per nucleus in the cells exposed to different types of radiation regimes. So this is how the setup looks for the flash radiation experiment. We have an electron source of radiation that is hitting the monolayer of cells, which consists of the co-culture of A375 melanoma cells and normal human melanocytes. And after the exposure to radiation, we can quantify and evaluate the dose that the cells were exposed to by developing a GAF-chromic film. A similar experiment is performed in parallel using an X-ray source. And by using a rotating X-ray chopper, we can obtain short pulses of the beam of X-ray that is hitting the monolayer of cells in a T-shaped signature. We can then compare the preview obtained using a TissueFAXS instrument with a GAF-chromic film that shows the exact heat map of exposure to the radiation, and compare the profile of cells that are expressing phospho-γH2AX foci with the intensity profile of the dose that they were exposed to. So as you can observe here, there are more cells that express gamma H2AX in their nuclei in the top and center areas of the slide flask, while the lower part of the flask was not exposed to higher doses, and you can see very few and very small foci inside the nucleus of the cells that are in this part of the slide flask. As controls, we have used the X-ray irradiated sample, as well as samples that were transported to the radiation facilities as environmental controls, but were not irradiated. Also, an untreated sample that stayed in the incubator during the experimental time. Just a reminder to the users of TissueFAXS, so we can scan the slides with different magnifications. And this is an example of a cover slip with cells that were labeled with antibodies against gamma H2AX and hexed to label the nuclei. We can see the entire region overview of the cover slip that is obtained using the 20x objective. If we click on a field of view, we can have a detailed view of the cells, but still we cannot quantify exactly the number of foci per nucleus with this magnification. Therefore, we need to scan shorter regions of interest with an increased magnification using a 63x objective and oil immersion. And you can see here an example of cells that were either untreated or treated with cisplatin, which is a chemostatic drug that has a DNA damage inducing capacity similar to the radiation. And therefore, we have set up our assays using this positive control. In order to start the analysis, we have to use TissueQuest. And for this, you have to open the software and choose Open Existing Project to import all the images for your samples to be analyzed. From this list of samples, you can choose a set of regions that you want to further analyze and for which you want to quantify the exact number of foci per nucleus. By default, when the TissueQuest software is started, you already have in the markers list DAPI for nucleus and TEXA as the channel where your signal for gamma H2AX will be quantified, because these are the two channels that were used for the scanning. For adding the dots analysis, you have to click on Add Dots Virtual Marker, and then here you have the option to choose a Texas Dots on DAPI new virtual marker in order to be able to quantify the dots and to segment them on the surface of the nucleus. And now you can see that this virtual channel is added to the list of markers that are quantified. And by default, you have to choose the various shapes as segmentation method and select only events entirely in ROI to proceed with the analysis. You can then click Next, and you obtained a new window where you have to choose measured parameters. Here you can see the markers that appeared on the first window, DAPI, TEXA, and TEXA dots on DAPI virtual channel. And for each of these markers, you can click on several parameters that can be quantified by the algorithms. For the analysis of gamma H2AX foci, we have chosen compactness and eccentricity along with the area and mean intensity for the nucleus. For TEXA's red, we have chosen mean intensity. And for TEXA dots on DAPI, we need to use the dots count parameters in order to obtain the values for each nucleus. So in the lower part of this window, you have a list of all the scattergrams and histograms that are generated automatically by the software, allowing you to quantify all the combinations between these parameters that are chosen in the Choose Measured Parameters window. This list can be can be modified at any time, even during analysis. When you press Finish, the software opens the project, and you can see on the left screen of your workstation the exact list of all the samples that are in the project, and you can click on a positive control. I have chosen here as an example the co-culture treated with cisplatin, and the images were obtained with 63x magnification, to show you that when you click on each of the sample, a cache is built for them one by one. After clicking all the regions in the list, you can then go ahead and start the actual image segmentation. Here is a print screen of the two screens of your TissueFAXS workstation, where you can see in the left part, you have the menu related to the list of samples in your project, and an image of one of the samples that is selected. And on the right side, there is a panel where you can modify every parameter for detecting the signal with high precision and increasing the signal to noise ratio detection in order to have a good image segmentation. On the right part, you have the scattergrams that are similar to the ones in flow cytometry, that are inspired by flow cytometry. And here, after the analysis of every sample, you will see dots that appear and are corresponding to values of your cells on each of the parameter that is analyzed. Clicking on this rectangle, you can then generate a new region of interest, a smaller one, to use it as a setup region. Because if you use the entire surface area of your sample, the algorithm will run many minutes, and it will take a long time to do the initial setup for your analysis. So here you can see the three markers that are going to be analyzed in this example: DAPI for the nucleus, Texas red for the nuclear signal for gamma H2AX, and Texa dots on DAPI to measure the foci. You can rename this new ROI by naming it setup region, and perform the setting up of the parameters on this smaller positive control. You should choose this region of interest, taking into account that the sample is heterogeneous, and within this smaller region you have to have nuclei that have also high intensity, as well as nuclei with low intensity, to capture all the diversity in your sample. Here you can see a magnification of this input window, and usually we can start by modifying nuclei size until the mask that is generated by the software overlaps perfectly to the nuclei size that we see by eye. And as an example, for our analysis, we obtained these shapes by using nuclei size of 40, and we modified the threshold range to be within 12 to 80 in order to have a good detection of the nuclei from the background. After detecting the nuclei based on the signal obtained on the DAPI channel, we next go to Texas shades overlay to modify the parameters for gamma H2AX detection. Here, you have to change usering mask to no, and choose the nuclei mask for detecting the signal. Then you have to optimize the background threshold depending on the characteristics of your sample. And we obtained a good mask using the background threshold of 5 for our images. Next, we can switch to Texa dots on DAPI and the image on Texa dots on DAPI shades overlay to detect the exact foci that are visible inside the nucleus. And we obtain the best results by selecting for dot size profile the middle intensity dots low background option. All throughout this procedure, it is very good to use backward and forward gating options that are in the TissueQuest software to optimize the exact threshold for each and every parameter in your list. Here, I'm showing you an example where we selected the lower part of the Texa dots on DAPI parameter to show the cells, the nuclei that have a very low to negative signal for Texas red. So if you press on this scattergram and choose see backward gating for lower quadrants, then the software will show you in red those nuclei that are negative for the parameter on the OY axis. Next, you can also choose forward gating by clicking on a specific nucleus and then going to the scattergrams and looking on the values and see if the nucleus is seen as a positive or negative event. Also in the histogram, this nucleus is seen at the mean intensity value corresponding to the Texas red signal inside the nucleus. As I said before, we used cisplatin as a positive control for DNA damage inducing agent. And with these samples, we have set up the gating strategy for our analysis. For the first scattergram, we chose DAPI mean intensity and DAPI area to detect single cells. We have set up the thresholds for every parameter, and also we have encircled the cells that correspond to single nuclei in our samples. From these events, we have then selected compact nuclei by choosing on the OY axis of the next scattergram DAPI compactness. This parameter helps us to remove from the analyzed events those that represent a fraction of nuclei or artifacts. So we have here only intact nuclear shaped events that goes towards the next step of the analysis. Because we work with a co-culture, we can use this cytometry gating strategy to separate our population of cells in cells with elongated nuclei that we know these are the normal melanocytes, and in cells that have non-elongated nuclei that are more round shaped, and these are the A375 melanoma cells. And after gating on a specific subset of cells, we can then measure the exact mean intensity of the gamma H2AX signal or determine the dots on DAPI to count the exact number of foci per nucleus in each of the subpopulation of cells. But also we can look at the total number of foci per nucleus in the entire population of cells that were exposed to different treatments. And here is an example of how you can use the backward gating and of TissueQuest to check the exact corresponding subpopulation from the gates that were set up and how they are exemplified, how they are identified by the TissueQuest software. So here is an example of a melanocyte that is in the elongated cells gate, and you can see here that indeed the nucleus is elongated and is smaller than the one that is in green and corresponds to A375 melanoma cell. And here also you can see the gate for non-elongated cells that correspond to all the events that are the melanoma cells. You can also set up thresholds on the TexaDots-on-DAPI channel based on the baseline intensity of the gamma H2AX signal in non-irradiated cells. Also, in our organisms, in cells, there is a baseline expression of gamma H2AX, although the cells are not exposed to any type of treatment due to intrinsic metabolism that generates stress on the DNA. So in our example, we have compared samples obtained from the two experimental flasks exposed to the 1 petawatt laser generated electrons in comparison with the cells that were irradiated by a conventional X-ray source. In order to be able to obtain a statistical significance comparison data, we have to export the numerical values from the TissueQuest in order to be able to import them in other softwares like GraphPad to be able to generate a graph of the data and to measure statistical significance. For this, you have to generate the raw data from every sample that is analyzed and then filter the events based on DAPI compactness and DAPI eccentricity to look at number of foci per nucleus, either in a subset population like melanoma cells or in the overall population of cells that were exposed to radiation. So we have filtered the DAPI compactness, based on DAPI compactness, the events that were analyzed. And we used the previously generated threshold of 0.73 to select for cells that are compact. Further, we have then filtered the cells based on DAPI eccentricity to determine the cells that are have either round-shaped nuclei or that are more elongated. And then with this exported Excel list, we have opened the GraphPad software and chose a column graphing method to compare the treated samples and to untreated controls, and to obtain such bar graphs that can be further modified to generate colored graphics and to label them based on the treatments the cells were exposed to. At the end, we obtained results such as the one shown here, where you can look at the effect of radiation on the whole sample, on the co-culture, and also separately to look at the effect of radiation in cancer cells versus normal melanocytes. So after this experiment, we were happy to see that for a certain region in the slide flask, we obtain indeed a differential effect where the cancer cells had shown a higher number of phosphogamma H2AX foci per nucleus, whereas the normal melanocytes were not affected by the treatment and they show a similar number of foci with the cells that were not exposed to radiation. Here is a table with the number of events that were analyzed for each and every treatment. And you can observe that we have analyzed thousands of cells as compared to other analysis softwares that allow you to only use single fields of view from your sample and have only low statistical power. You can also use violin plots, but you have to pay attention that if the number of events that you use differ a lot between your samples, or if the number of cells in your co-culture is very different. And in our case, the melanocytes are 10 times in lower number than the melanoma cells. So you have to be very careful because these bulky violin plots can mistakenly be interpreted as a higher signal, but instead there is a higher number of events that is analyzed. So just be careful when you choose the exact graphing option for your data. So in conclusion, using TissueQuest, we have successfully validated the DNA damage-inducing effect of laser-induced very high energy electron irradiation produced by the petawatt facility in Chetal, and also ultra-high dose rate exposure of melanoma melanocyte co-cultures provides evidence of a possible differential response and a flash-sparing effect on normal cells. But we still have a lot of work to do because it is necessary to uncover the flash-induced molecular mechanism of anti-tumor action. Most of the experiments were done in animal models, but there is no deep study on the molecular effects of flash radiation in cells. And also, there is further work to do for preclinical testing of selected doses of radiation that could be translated to clinical application. And we have now a larger team and a new funded project that aims to characterize the effect of flash radiation in the tumor microenvironment. So we'll look at ovarian cancer and melanoma and different subsets of immune cells, such as macrophages and T cells, perform proteomics to look at the secretome of cancer cells exposed to electron flash. And also we benefit from our advisor, my mentor from the postdoc, Dr. Daniela Matei, an oncologist in the OBGYN at Northwestern. And I encourage you all to send me an email whether you have questions or problems related to TissueQuest analysis of gamma-H2AX foci in your specific experiments. Thank you for your attention.